What is up, guys? In this video, I'm going to be talking about B cells and how B cells respond to pathogens like viruses or bacteria or fungi or parasites that are inside of your body. And it is a very interesting and nuanced topic, but I'm going to do my best to keep this at a pretty high level so that you don't need too much biology background to follow along. And so before I get too deep into B cells explicitly, I want to focus a little bit on the overall job of your immune system. And your immune system has the role of identifying your self versus your non-self. And self refers to your actual human cells, and then your non-self refers to pretty much everything else in your body, like bacteria or E. coli. And so this is called specificity. And you do need a very specific immune response, because when you lose your specificity, that is when your immune system starts attacking yourself, and you essentially have a civil war going on inside of your body. And this is seen in women, particularly in their later years with arthritis, in which their immune system starts attacking cartilage and creating inflammation and attacking their own joints. And so the immune system is very powerful at not only defending yourself, but also it can be misappropriated and start attacking your own cells, which is very bad. So knowing this process very well is extremely important. And the purpose of this kind of lecture series is going to be diving into how scientists and engineers are able to start making use of these B cells in labs in order to start doing things like making cancer therapies from it. And so that'll be a future video to this, where we can start actually deriving utility from our knowledge of biology, which I think is pretty cool. And so to begin with here, I have to the right a naive B cell. And this word naive refers to the fact that it's pretty undifferentiated yet. It hasn't really been activated, and there's a lot still to come in this story. So this is kind of the opening scene to a play that is happening inside of your blood right now. And there are millions and billions and trillions of pathogens out there in this world, especially in the air that you're breathing right now. And so your body needs to be able to respond and react to these pathogens. And so to give this story a plot, I'm going to introduce a virus. And what will happen is this virus will have some kind of extracellular features to it. And a virus isn't necessarily a cell, but it will have some features on the outside of it. These features are some form of protein generally. So viruses have capsids and these other things that help them latch onto cells that they're about to infect. And so the purpose, or the main point here, is that our virus will have things on its outside that can be recognized. And so I want to hit home specificity here, because specificity in your immune system is achieved in B cells by having extremely specific, highly specialized interactions between two things. And so B cells are very unique in that they have these surface receptors on them that are specific for one kind of protein on a pathogen. And these things on our pathogens, these proteins, we commonly will refer to as epitopes. So epi from the Greek meaning above. This is referring to, it's kind of like the skin of your virus will have this unique character to it, this unique shape. And you're going to have these B cells roaming around inside of your blood. And when they encounter an epitope, they may or may not have an affinity for it. And so what we're going to find time and again is that most of your B cells will have no affinity at all for the epitope on the pathogen that is inside of your blood. So, you know, even though you might say, oh, wow, I'm really impressed, the B cell has encountered a virus and I can now have an immune response so I don't get the flu this year. That's not necessarily the case, because that B cell needs to have a very specific variable region that will have an affinity for the epitope on your pathogen. And so this variable region, so I'll just circle one of these things here, these variable regions on the surface of your B cell I believe they're also called B cell receptors, BCR, are very specific. So it's going to be a theme we keep seeing time and again here. But these B cell receptors are going to have some kind of specificity for the epitope on a virus, for instance. And so assuming that the B cell receptor has an affinity for the epitope of a pathogen, the next thing that will happen is that your B cell will become activated. So the key thing to take here is if the BCR binds to an epitope, which is on your virus or on an E. coli or on a yeast or any non-self cell in the ideal situation, your B cell is activated. And so what does it mean when this B cell becomes activated? Well, because of you will now want to scale up your immune response, because if you just encountered one virus in your blood, it's very likely that there might be millions or billions of other viruses nearby of that very same type, because we know that viruses grow and expand so quickly that your immune system will need to respond to that at a similar scale. And so this activated B cell, I'll try to write clearly. This activated B cell will move from being a naive B cell into the activated B cell form. So we're going to move from here to here, and a lot of internal things are going to happen with inside the cell that are going to be causing a lot of eukaryotic genome expressions to happen. So this B cell is going to start doing a lot of things and kind of shaking up in anticipation to do a lot of other stuff. And the key thing to take away from the life cycle of a B cell, without getting too deep, is that what we're going to get out of an activated B cell are effector cells and memory cells. And the purpose of our effector cells, our effector B cells, and these are also called plasma B cells. Sorry, hopefully we can understand. These are called plasma B cells. Their job is to crank out antibodies. And just to review, our antibodies look in the shape of a Y. They have a heavy chain and a light chain. And these antibodies are going to bind onto our virus or whatever pathogen activated our B cell in the first place, so that we can have macrophages and neutrophils and all these other cells within your immune system respond to it. And if we now look to what our memory B cells will do, so essentially this B cell will undergo a lot of mitotic divisions, and they're going to be expressing different genes to make it an effector cell or a memory cell. The memory B cell will go on to have the job of remembering the pathogen, and this serves for the purpose of your secondary immune response. So that if you encounter that virus at a separate time in a year or 2 years or 5 years from now, your memory B cells are going to be able to scale up an immune response very quickly thereafter, so that you don't get the flu that year, or if you do, the symptoms will be a lot less severe. And so if you wonder why you get your flu shot every year, your flu shot is essentially a castrated or very weakened version of the virus that researchers at Pfizer or whoever have guessed will be this year's current flu. And they give you a weakened version of this pathogen in order to make memory B cells, so that when you are exposed to the real virus, you will be able to scale up an immune response a lot faster, because you won't need to go through this whole cascade again of a naive B cell getting activated and then forming the actual memory cells. You will already have the memory B cells present in order to respond to the pathogen. And to wrap up what's going on with our effector or plasma B cells, these are going to be cranking out tons of antibodies, And these antibodies are going to stick onto the surface of our virus or the other virus's progeny that already exists within your blood, because chances are that virus has already infected one host cell and a million other viruses now exist within your blood. These antibodies that are being cranked out by your effector B cells are going to be sticking onto those millions of other viruses that are present in your blood, so that they can very quickly be recognized by your immune system, and they also interfere with the virus's ability to infect other host cells. So this is a high-level overview of what's going on with your B cell immune response. I hope you guys find it useful, and thanks for watching.